I'm Meredith Blackwell, and this is Don Pfister, and we've been interviewing people for the Oral History of Mycology project. We're on the University of Florida campus at the Edge in the hotel, the Hilton Hotel, and we are at the Mycological Society of America meeting, our annual meeting, and today is the 13th of July, 2022. So Don Pfister, who's at Harvard. He's just retired. I'm retired from LSU about 5 or 6 years. And Nemat Keyhani is here at the University of Florida. And we'll start out with asking him, where were you born? What's your background? Something about your family? Did they encourage you? How did you become interested in fungi?
Oh, well, all right. So I was born in Brussels, Belgium. My father is Iranian and my mother's Belgian. My father went to medical school in Brussels many a year ago, met my mother, and had a bunch of kids, I being one of them. And we moved to the States when I was around 5 or so. And we lived in Philadelphia. Mostly I grew up in Philly, spent a couple years in Memphis, Tennessee. My parents were at St. Jude's Research Hospital there. And then back to Philly and around. We traveled around a bit. And then I did my undergrad in Philadelphia at the University of Pennsylvania. And then did my graduate work at Hopkins in Baltimore. And my graduate work and even postdoc was nothing to do with mycology or entomology.
What were your majors in school? Were they biology?
So as undergraduate, I was biochemistry and sort of liberal arts, a mixture. At UPenn at the time, and maybe still, you could sort of make your own major if you had a lot of credits and a lot of different things. So I kind of had something like that. And I worked in a research lab as an undergraduate, and I think that was important in sort of stimulating my interest in doing research. I sort of knew early on, well, both my parents were researchers, I should say that.
And your mother's a physician?
My mother has a PhD in chemistry.
Oh, okay.
And my father has an MD, but he never practiced medicine. He wanted to do research, and so he always did research his whole life. So I was sort of a faculty brat. And so I sort of knew the milieu of the university, and it was sort of a natural kind of thing for me. And I enjoyed research. I enjoyed, this kind of scientific inquiry. And so, yeah, I decided to do that. It was either that or art school, actually. And but art school, well, even now in grad school in the STEM, they pay you to go. Art school, you have to pay to go.
Yeah.
That was a big. I think if I hadn't gotten into Hopkins, I probably would have gone to art school. But I was like, I can't turn down Hopkins.
And what were you doing there? What was your research there?
So my work there was on marine bacteria. My mentor was a wonderful person by the name of Saul Roseman, and he studied the phosphotransferase system in bacteria, this sugar uptake and metabolism system. And so I began working on a project looking at marine bacteria, how they degrade crab shells, shrimp shells, and
and probably fungi too.
Well, I didn't study fungi.
No, but I was just wondering, are they the same, the chitinase? Did that chitinase?
So the transition to working with fungi stems from that. When I thought, naively enough at the time when I thought, okay, I have a background in chitinases, degrading, bacteria degrading crab shells, which have chitin. Why not start studying fungi that degrade insects? They also must have chitinases. And of course, the systems are very, very different and things like that. But nonetheless, that was part of the transition.
Okay.
Yeah.
Yeah, so when did you make the transition?
Well, for me, it's an interesting background story is that when I was a grad student, I read this book called Mr. Wilson's Cabinet of Wonder
Yes.
by Lawrence Weschler, I think.
Yeah.
And in it, he talks about this Museum of Jurassic Technology. I don't know if you've heard of it. And obviously, a lot of it is sort of made up stuff in this museum. It's very fascinating. I think it's called like the Natural Unhistory Museum because it has curiosities and these things like that. In this small little book, there's a story called Inhaling the Spore. And what it is, it's a story about the Cameroonian stink ant, which is not a real ant, but it's a made-up story. The Cameroonian stink ant, as the story goes, is one of the largest ants in the world. And it's so large that it can make a cry that's audible to the human ear. This is not true, by the way, but this is in the story.
Give me your disclaimer.
Yeah, but it's a very fascinating story already, right?
Yeah.
And normally it forages on the floor, the canopy of the jungles. But occasionally it begins to elicit this sort of very erratic behavior. And what it does is, instead of staying on the floor, it has this phenomenon called gravitotropism, where it seeks heights. I think you guys know where I'm kind of going in this. But it seeks heights, and at a certain point, it climbs up a stem, a leaf, a tree, or something like that. It fixes its mandibles on there, and out from its head grows the fungus. And then the fungus sporulates and rains its spores down to infect new unsuspecting ants. Well, this is the sort of zombie ant phenomenon, that sort of has captured at least some people's imaginations. It's based on a true phenomenon, but this particular story is not true. And I think the genera of fungus that they mentioned is just a made-up one. So now we know it's these.
This.
Yeah, it's a very thin book, and so now the phenomenon is these Ophiocordyceps that infect ants and basically do this.
And there's now a zombie stink bug.
Yeah. Oh.
From Costa Rica, and we were just kind of talking about it, but Bill Everhard had a class that collected it, and they kept finding this, and so I got involved with a student with it.
There's probably a lot of this going on.
Yeah.
There's probably quite the diversity of these kinds of manipulations.
All kinds of things.
So that stimulated you to look at other systems.
Yeah, so I said to myself, if I ever am lucky enough to get a faculty position, that's what I want to study. And so sure enough, one day UF offered me a job. I'm not sure why, but they offered me a job. And
as a bacteriologist to study
microbiology. I'm in the microbiology department, and back then things are a little bit different. I mean, I've been here about 20 years. They didn't quite ask me what I wanted to do.
You didn't have to give a vision.
I didn't have to. Yeah, not like I didn't have to have everything mapped out, like a grant ready to go and all of that kind of stuff. I had ideas, obviously, but today they expect you to have, things already half done. I didn't do that, and so I said, you know what, I'm going to try this. I tried a few other projects, but this was the first one that I got funding for, is to study insect pathogenic fungi. Okay, so that's what I set out to study, is insect pathogenic fungi. And I got a slim 2-year NSF grant, not even a full grant. I had put in a full grant and they said, we like it, but we don't like it enough to give you all 4 years. We'll give you 2 years. How about that? I said, of course, thank you. And that started it. And so what I started studying are these insect pathogenic fungi. And the question is, for me, was at that time, it was pretty much a black box of how they did what they did, the biochemistry and the genetics behind it. These organisms had been known for well over a century, and people are interested in them for biological control of
Were they
pests.
Bavarias?
And I started studying Beauveria, Beauveria bassiana, named after the Italian scientist Agostino Bassi. When I read about him, I said, I know this is the project that I want to work on. He was a very interesting character.
Expand on that a bit. he was, what, early 19th century, wasn't he? 1830s, 40s?
Yeah, so he was born in 1773,
Oh, even
and
before
he
that,
he died in 1856, and he was the son of sort of these landed Italian aristocrats, and he became sort of a Renaissance man. He was a diplomat, he was a writer, he was also interested in science. According to the biography I read of him, his parents didn't really want him to do science. They wanted him to manage the estates and everything like that. But he had this sort of interest in scientific and scientific agriculture. And what had happened is that at that time in Italy, they had a thriving silkworm industry. So a few centuries before, Italians had gone to China and there was a silk trade, and they said, well, why don't we just kind of steal some of these silkworms and bring them and make them ourselves? And that's indeed what they had done. But periodically, these sort of silkworm farms would be devastated by a disease called white muscardine disease, Blanca muscardino disease, and which turned the caterpillars into these candy-looking like things, which is not white and fluffy. I mean, by today we look at that and we say, oh, it's a fungus. But back then, obviously, they didn't quite know that. And what he did was a very simple and ingenious experiment. You know, I think everybody can appreciate this, is he took a dead insect and he pricked it with a needle, and then he took that needle and pricked a live one. And he showed that that live one then develops disease. I mean, it was really something very revolutionary at the time. Today, it seems simple. And so he developed basically the first germ theory of disease. Pasteur was later, actually. Pasteur was, I think, born in 1822. And certainly knew about Agostino's work, and this was sort of part of his contribution. He did other, and he, by the way, made a very simple recommendation to the farmers of silkworms. He said, if you see one of these things, remove it immediately from the rest of them. I mean, it seems like a very simple kind of thing, but it was effective and it worked.
Yeah.
Yeah.
Yeah. So.
No idea about disease transmission. Control.
But he figured it out.
He figured it out by a very simple kind of experiment. You know, I mean, he did more, obviously, but and so much later, when they found out that this was a fungus, they named it after him. So it's the fungus that I study mainly, or one of the fungi I study mainly, Bouveria bassiana, is named after Agostino Bassi.
So tell us exactly what you're doing these days then. Starting with those early projects with NSF funding for 2 years.
So that was on the Bouveria. So my lab, we study two basic kinds of fungi. One are fungi that are pathogens of insects, and the other, it's a more recent project, is fungi that are symbionts of insects, sort of the other side of the coin. One is very harmful to the insect, and the other one is very beneficial to the insect.
And by the way, you had two students. Each one had one of those types of projects,
yes.
And they were back to back on the same bulletin board.
Yes.
Yeah, very nice students.
Yes. So the Bouveria project, the insect pathogen project, I mean, our interest was to really understand how does this fungus, amongst the hundreds of thousands of fungi out there, which are non-pathogenic to insects, what has made this fungus a pathogen of insects? Adaptations, the genetic networks, the biochemistry involved. And obviously, we have not solved the whole issue, but we've made a significant progress. And obviously, I'm not the only person. There are many other people in the field who are studying this.
And the symbionts?
The symbionts is very interesting. So these are symbionts of beetles, and these are bark and ambrosia beetles. And these symbionts are, so the ambrosia beetles and the bark beetles they're tiny little creatures. I mean, you know, like you can put them on a penny and they're like a few, you know, half a centimeter or smaller. They bore into trees. They bore or bear into trees. I'm not sure what they bore into trees. And they make little tunnels, and then they lay their eggs, and the larvae develop in these beautiful galleries. And different beetle species have different shapes of galleries. It's kind of wonderful when you open them up and to look at them. Different architectures, just like ant mounds, you know, different architectures. And but they don't eat the wood or trees or anything. Instead, what they do is they release a fungus that grows on the tree substrates, and then they eat the larvae, and the adults eat the fungus. So they're farmers. They're basically farmers, fungal farmers, and this is, an interaction that's probably 300 million years old, that these guys have co-evolved to do it. And there's a huge diversity of beetle species that does this, and it appears as if this adaptation of fungal farming has evolved numerous times independently.
In the beetles.
In different beetles. So obviously it's something that's very adaptive for them and has a lot of selection for it.
Is there a lot of difference in the fungi?
Yes, so the fungi, you know, across these different beetle genera that have this are completely different. And there are, not only are they different, but so what the beetles do is they have evolved specialized structures to carry the fungi. The mycangia, and these are called mycangia. And different beetle genera have completely different mycangia, architecture, even location on their body. So the ones we study are beetles that have what are called suboral mycangia. It's sort of right around the mouth parts inside the head. But there are other beetle species that have it on their back, dorsal mycangia, and in different areas. So it's an incredible diversity of interactions.
Are you looking at one
We are looking at, yes, ambrosia beetles and their interactions with this sort of group of fungi known as Raphaelia. Now they've been renamed Harringtonia. And these are suboral mycangia, and they are considered promiscuous. So there are some mycangia that are one fungal species, one beetle species. It's a very tight, you know, specificity. These guys, they can take any number of a collection of related fungi. It's not like they can take any fungus, but they're all related, but they appear to have a more kind of leeway in terms of which fungus can occupy and colonize their mycangia.
Now, I'm really glad to see that name because I know Tom Harrington.
Oh.
And when he first discovered this fungus, I guess he worked with someone here in Florida. But he was really interested in it because it was a very recent introduction.
Yes.
And it's very specific for the family of plants that it will infect. And so I was actually able to review the paper really fast for him. He wanted to get it out there with the description of the species.
Oh, wonderful.
Yeah, so it was really quite interesting. And so I've sort of followed the story, but it's really neat. So I'm glad you've named it for Tom or someone has.
Yeah, not me,
Yes.
but they have, yes. Yeah, it's so the story, it is an invasive species to the United States. The beetle came around maybe the turn of the century, 2002, somewhere in Georgia. I think they quite know.
Savannah, I think.
Yeah, and the beetle species, it's a Xyloborus glabratus. There are indigenous Xyloborus species here, but this is an invasive, and it brought with it its own collection of these fungi. And most of these fungi are benign to trees. You know, they make a little bit of damage, but they don't really do harmful. But this invasive beetle brought with it one particular fungal species, which is now called Harringtonia loricola, which is a devastating pathogen to the trees here in the United States, the naive trees. It's not a pathogen in Asia, or at least not a significant pathogen in Asia, but here it's become a very devastating disease of Lauraceae family
And
trees.
I was wondering, has that got into the avocados?
Yes.
Yeah.
Yes. So avocado is a member of this family, and the beetle has expanded, in its invasion. It's reached South Florida, and it is devastating and killing avocado in Florida, South Florida. And it's moving westward. And there is no treatment or cure. You have to cut the tree and burn it. And unfortunately, it can kill healthy trees in anywhere from 3 to 6 months.
And I remember Tom was thinking that, well, avocados are our largest crop, the economically important member of the Lauraceae, but he was worried that it might eventually go around the Gulf and get to Mexico, where there are other crops that would be involved.
I think it's only a matter of time. I think and California too, there's a big barrier, but, it's by trade. Things move, cars and airplane and people. And so I think it's really just a matter of time before the beetle. And the other thing is that there's now definitely evidence that the Loricola or whatever can switch hosts to indigenous beetles. And that's one thing we're sort of studying. So that even expands further its potential range. If it's now being carried by indigenous beetles, then it's really just a matter of time.
Stephen Kassar, who worked on these beetles very early when DNA was first being used, to distinguish them, because the fungi that they carry are very difficult to distinguish from the cultures, I think. But anyway, we were concerned that one of the native beetles, might be able to carry one of the blights of oak trees, and so we hope that hadn't happened. I haven't heard that it has. But anyway.
So I think that's also a bark beetle vectored, and there's a number of examples of this sort of beetle vectored plant pathogens. And in South Africa, there's a very devastating one. There's one I think in California and in Europe and everything like that. Yeah.
And it's interesting. Many of these fungi we never know about until they get transported, and the trees are all naive, and then we recognize the disease. But the one, the Harringtonia, was not known until it came over here.
Yeah, absolutely.
Where I live in South Carolina, there's some butterflies that rely on some of these plants that it infects, and so there's been concern about that. They're butterflies that mostly eat these Lauraceae.
Well, supposedly, more than 500 million trees have been killed in the United States by this.
It's hard to imagine.
Yes.
And one more thing I want to ask, it's changing the subject a bit, but you're a two-scientist family, right?
Yes.
And has your wife been able to work here at Florida?
Yes, she does. So she started, actually, she has an interesting trajectory. She started in plant pathology, studying the citrus diseases of citrus, bacterial diseases of citrus. Then she switched to more kind of hardcore microbiology and studying tRNAs and tRNA structure and function. And now she's moved over to the kinesiology department, applied kinesiology, where she's using her genetic background in order to look at movement disorders in humans.
That's quite a transition.
Yes.
Yeah.
Yes, and she absolutely loves it.
And she's found her niche, and it's in a different department from yours, which is important. Yeah.
Different college. And it's sort of very interesting. But she sort of likes the more apply in human interactions, and it's certainly very important because it's motor diseases, aging, and these kinds of things. And the sort of teasing out the genetics of this kind of things and is kind of an interesting thing.
No, it's
Very difficult.
curious transitions. Yeah.
Yeah, it's the way we have to work sometimes, make these transitions.
Yeah.
Absolutely.
Yeah. Well,
Good. Well, thank you. This has been really
yeah,
Oh, it's my
interesting,
pleasure.
and we've learned a lot about these fungi,
Yeah.
so that's fun.
Yeah. No, that's great.
Yeah,
Good.
you must teach a lot.
I teach genetics and epigenetics. Yes, I